Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our final talk of the day to improve the selectivity and sensitivity of chimeric T-cell receptors for treating solid tumors. I'm pleased to welcome our speaker, Dr. Chun Wu. Dr. Wu is a tenured associate professor with a joint appointment in the departments of chemistry and biochemistry and the biological biomedical sciences at Rowan University. He obtained his PhD from the University of Delaware, where he worked on protein force field development and conducted molecular dynamic simulations of amyloidogenic peptides. His lab's developing a novel evolution theory, not only to best explain the molecular evolution of pathogens, but also to accurately identify hotspots in their genome for developing first/next generation vaccines and drugs to treat these infectious diseases. A very warm welcome to you, Dr. Wu. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Wu's presentation. You can also benefit from some fantastic supporting literature, which you can download for free below the video player. And now, without further ado, I would like to hand over to Dr. Wu to begin his presentation.
Thank you, George, for that nice introduction. Okay, today's talk, I will cover the general introduction on the computational approach to assistant drug discovery. And I will specifically cover the overall of the computer-aided drug design strategy, and then specific technique, including the homology modeling, molecular docking, and in particular, molecular dynamic simulation. And then I will use two examples. And first is a small molecule drug discovery. And second, that's our central topic, is how to optimize the chimeric T-cell receptor. Okay, let's start the journey. So why we want to do the computer-aided drug design? Because we don't want traditional blind testing approach, which will taking too much time and cost for development. We want to fully use the available information about the drug target, about its sequence, about its structure, and even its dynamics, all the property to do the virtual screening in the computer. So we can narrow down to the good candidate, small list of good candidates. So here I show you a workflow commonly used in the drug discovery. So we typically will start from the drug target structure, and we have the library of the compound. In this case, is 17 million compounds. And then we do some filtering based on predict drug property. And then we typically do some docking to get some docking score, docking the compound to the active site and ranking that. And then we do clustering of the candidate to diversify the molecular scaffold for candidate. And then we pick compound, and then that smallest compound can subject to the experimental testing. So by this way, we fully incorporate the computation into the workflow of drug discovery. Okay, so first we need to get the structure. So if there are experimental structure, it's fantastic, great. But in some case, the structure not available, then we can use a technique called homology modeling to build the structure. The idea is very simple. The protein has a similar sequence, will have a similar structure. So if we can find the sequence similar to our target sequence, which already had a structure, then we can use that structure as template to build the structure model we want. Even better, you probably heard of the AlphaFold from the Google Deep Learning program. Now they're able to make a very good prediction on the other part of the protein structure. So that even give us the more structure for the drug-based drug discovery. Okay, so with the structure, then we want to identify the critical part of the drug binding site that typical are the active site. For the enzyme, that's basically substrate binding site, typical are the good place for testing. And then we can dock the compound into the active site and the ranking to guard fitness, and we call the docking score. And then we can ranking the docking score to guard top compound. So that's allow us to screening large number of compounds. So in my laboratory, typical one day we can screening the millions, 100 millions compounds to a particular target. Okay, and another fantastic tool which I emphasize in this talk is the physics-based molecular dynamic simulation. AlphaFold can make very good prediction on the order part structure based on the solved structure. But we know important part of the protein has a disorder part, which are flexible part, don't have the structure solved, and because it's intrinsic disorder and it has very dynamics, but their function important. And just like our body, many parts are rigid, our head are rigid, and our leg and hand are rigid. But we know we need a joint, flexible part in order for our body to fully functional. For protein, is the same way. You need rigid part, but you need flexible part. And how to get information about the flexible part? Well, the physics-based model offers such kind of a great opportunity, and I will demonstrate you. Okay, so for the physics-based model, it's based on physics. So like here, the transformer go from the human shape to the car, basically fades to different function. Okay, car is good for running, and the man shape is good for fighting. And we know underlying the shape change is due to the physical interaction, the force. So when you have the force will generate acceleration will change the speed will move the position. So then you got full motion. This idea can transfer to the molecular system. And if we define the atomic interaction, so like the chemical bond angle, dihedral angle, and long distance interaction, like charge-charge interaction, like dispersion interaction, the van der Waals interaction. If we develop the parameters to characterize those interactions, then we can simulate the motion of the biomolecule. Okay, for example, here I start from an extended peptide, and this peptide has been shown is a fold into the helical structure. So then we can see whether it can fold into the helical structure. Okay, it starts the folding process, and it's forming the backbone main chain hydrogen bond. So now you see it fold into the helical structure. And that's kind of the physics-based model, and which has the advantage not depend on the database. And it's global model, so any sequence you can throw in it, even though it has some approximation, but it can give us a lot of the information. Okay, so for the molecular dynamics simulation, the time step are critical because we are limited by the fastest motion of molecular system, which are the vibration. So that basically means the longest time step for our integration Newton's equation, we only can pick 2 femtoseconds, which are the period of the hydrogen vibration. Okay, and then in the real folding process, it has a different time scale. So for the rotation of the side chain, it's probably picosecond. And for some loop closure, it needs nanosecond. For helices formation, it's kind of the 100 nanosecond. For the helping, you probably need a microsecond. And for folding some of the slow folding protein, you probably need a millisecond. And then for forming some of the aggregates like the aminoid fiber is probably need months or years. So time scale is important to see the phenomenon interested in. And for that, we need computing power. Okay, so here's a brief kind of the review, the development of molecular dynamic simulation. So it started from the 77 with the Apple computer. So only we can simulate it small number of particle and integrate very short period, and then in gas phase. And Dr. Wang is my PhD advisor, so he's fortunate to use the supercomputer to simulate the folding small protein in the water box for much longer time, even though it's still not fold to the native structure, but it's a milestone. That work inspired Dr. Xiang, who invests money to develop specialized chip, speed up the calculation. So you see back to the 2010, with the computer he developed, it's called the Anton, and Anton is the scientist invented the microscope. So he wanted to use this Anton machine to look the folding, to look the atomic structure of the protein. And you see it's able to fold into the native structure. And the simulation time now is go to the 100 microsecond. And now he has a second generation Anton computer. So actually our group has allocation on that. The work I'm going to discuss is use Anton 2 computer to do the simulation. Well, not every group has a chance to use the Anton 2, but the graphic card are available, and that's great resources. Actually can done a lot of good work. And then the computing power keep increasing. So you wait and you got the more powerful tool. And so here's the throughput for the Anton computer. So this compares the Anton 2 with the Anton first generation, and also compare with use the GPU. So like for example, here use the 1024 node of the existing CPU, and so you can say the throughput difference. Okay, so if you use a commercial hardware per day, you only can run 1.5 microsecond per day, and use Anton 1, so it's roughly tenfold kind of increase the speed. And if you use the Anton 2, well, it's a 40-fold increase for this small system. So the small system. But when you go to large system, okay, let's jump to the million atom. So million atom system, you literally can simulate all the protein, okay, typical protein, okay. And so if you use a commercial available CPU-based hardware, and you only can do 0.9, 0.1 microsecond per day, but you use Anton 2, you literally 95-fold speed up, so 100-fold speed up, so very fast. Okay, so let's show some the application of molecular dynamic simulation before we jump into the drug discovery use it. Okay, so here's what I did when I was postdoc, and I tried to see if use the physics-based model, we can fold some the small protein. So this protein has a helical part, has the helping part, and you kind of can see the helical part form first, partly involving the shorter range hydrogen bond, and the helping part involving the long range cross-strand hydrogen bond. So it takes a little bit longer time for search to the native structure. Okay, so now it's fold into the correct structure, and then you see the hydrophobic radio packing inside stabilize the tertiary structure. And this is another example to show how the drug interacting with its target, which are the DNA. And this is the first line leukemia chemical therapy drug, and it's working by insert between the two DNA strands. And we know DNA coding the genetic information, but the genetic information need to read out, you need to separate those two strands, and then based on base pairing to do replication or transcription. Okay, when the lock is not able to do that. But here, what we're interested in is how this drug insert here. And crystallographer solve this crystal structure, and they suggesting might be just the open up so drug can get in and stay there. And this work I did 10 years ago, I want to see if the physics-based model can bind to here. If so, how it go inside. Okay, so here's kind of the simulation result. And you kind of say first is diffusion process. Okay, solvent I have removed. And you say the base pairing is broken, and then drug starts insertion. So once insertion occur, and then you have the base pairing. So this is always called the flip insertion mechanism. So from the dynamic simulation, we kind of can quickly pick up. And what's to mention, so this work has cited over the 60 times. Okay, so in the drug discovery, small molecule drug discovery, 30% of the drug targeting the membrane receptor, which is a G protein copper receptor. And this is the example. And it works by sensing the extracellular information, pass that information inside the cell, so cell can do proper response. And it's a signal cycle is like this. So when you don't have the signaling, so G protein, which has a 3 unit, alpha, beta, gamma, will associate intracellular part, and then it has the GDP associated with alpha unit. And this is the inactive state. It's waiting there and to sensing the signal. So if you have the signal, we call the agonist or activate, and then it will trigger the conformational change. So this GTP will be exchanged for the GTP. Okay, GTP is triphosphate, GTP is diphosphate. Okay, so it will exchange. So then this G protein is activated, and then it will separate it and then send to the effector and then to activate downstream pathway. Okay, and once done its activation, it will do the hydrolysis. So GTP will go back to GDP. So now it will go back to the inactive state and associate with the apo form of the receptor. So that's what being suggested. Okay, so here the very interesting is how the signal transduce cross membrane to the inside, how the G protein know their signal binding outside. That's an interesting physical question. So for that, we can use molecular dynamic simulation to see how the signal can go inside. If we have good understanding, this might be help us to design the better drug. Okay, so here we try to control the system. So we already has the crystal structure of a G protein copper receptor with the G protein, and in here it has the activate. But amazingly for receptor, it responds differently if you have a inactivate, which we'll call the antagonist. Okay, so that basically it will not activate this G protein. Okay, so but in this structure, we don't have the GDP. Well, we have some structure G protein with the GTP. So you can imagine we can align this structure here and transfer this GDP to here. And then we can put a different kinds of ligand into the here. So without the ligand and with agonist, with antagonist, and we want to see if physical model can give us the proper response. Okay, so here's what I did. So I created that system, and I use a GPU to do the simulation. And so here's the GDP, here's receptor, that's membrane, extracellular water, inside water, and alpha beta unit. Okay, so here is the check. And we know without the signal, this GTP should be stay there. And let's see if it stays there. So this is the simulation. And I remove the lipids, remove the water to make the more clear for you. Okay. So yeah, it stays there. So that basically means that no signal should not be activated for this G protein. It should stay there. Okay, so now we can really testing our hypothesis if it works. So we put antagonist, which you know is inactivate. So inactivate, even you put ligand here, does not generate the proper conformational change and to induce leaving of this GTP. Let's see if that's true. Okay, yeah, it stays there. So now let's replace this into the agonist. So agonist will transduce a signal cross membrane to this side. And let's see. Okay, so it's kind of starts flexible and this GDP. Oh, okay, so it's leaving. So this means that this physical model do give us some the expected information, and then we have repeated this for many times. So statistically, it show yes, it's a follow our expectations. The work is published in the one. Okay, so now with some confidence on the molecular dynamic simulation on those computation tools, so now we look into the computer-aided design. So this project we just finished this spring, and it's literally targeting a enzyme which are the play important role in the epigenetics, which will good target for the cancer. And again, structure is available, and the reference the current BIDFS inhibitor is also available. So it's very good starting place. Okay, so we have this structure, we have this active site, and then we simply just docking 70 million compound to this site and do our virtual screening virtual flow, okay, which I have explained. But I adding one more step, okay, of the docking, of the filtering by drug property, and we do the induced filter docking, which is more accurate than the rigid body docking. And then we do the molecular dynamic simulation to check stability and then to calculate the bonding free energy. And with that, we picked 6 compounds. Okay, and then we want to do the wet lab testing, to do the enzyme inhibition assay, to do the cell viability assay to check if that works. Okay, so here is that 6 compound we identified, and then we do. And the amazing things, several compound has activity. Well, I cannot tell you which one. And the best compound actually show very good inhibition, which has the IC50. Okay, IC50 basically is the inhibition concentration at 50%. So this is the response curve. As we increase the drug concentration, we should see the reduce of the enzyme activity. So at 50%, that's our IC50. So IC50 is about 10 micromolar. Not a bad number. Okay. And then we do the cell viability, use the triple negative breast cancer cell line to do this one. And then we also got effective concentration at 50%, is 10 micromolar. This is a beat the number in the report in literature. So so this is still the ongoing project, and we plan to move this compound to the animal model. So what's the overall cost for this project? Okay, 6 compound is roughly each compound $60. And then this assay is about $1,000. So overall experimental part is less than 5,000. Well, this computation only take us several weeks. So I think this is the one example. Okay, so let's now move to the central topic for this project, for this talk, and it's concern the chimeric antigen receptor, which used in the T cell therapy. So the T cell therapy is basically taking the blood from the cancer patient, and this case is the leukemia, okay? And then you isolate the T cell, and then do the transfection, the gene coding the chimeric antigen receptor, and let express. So then this T cell will enable it to recognize the specific biomarker on the tumor, and to activate this T cell, start the killing program to remove the tumor. So this T cell can be grown in the large quantity outside the body and then inject back to the patient. Okay, and this leading to the cure in the cancer treatment. This is the Emily, this is 10 years ago, and he diagnosed leukemia. Well, you know, partly it's a sentence, the death. And fortunately, in the Philadelphia Children's Hospital, this treatment available developed by the professor from UPenn, University of Pennsylvania. Okay, so he did that one. And this is, she's a now and healthy. So it's kind of the cure for her. And so this revolution treatment has been proven for 6 products. Okay, and they are kind of the targeting the blood cancer. Okay, so people want to extend this method to the solid tumor, breast cancer, lung cancer, prostate cancer, or other solid cancer, but no success. And one of the big challenge is still lack the selectivity sensitivity, because for solid tumor, typical the biomarker on solid tumor also present in the normal cell. Okay, so you're killing it, you're also killing the good cell. And also the sensitivity, because in the solid tumor, the microenvironment is very different from normal tissue. So it's more acidic and lack of oxygen. So those environments makes the T cell less sensitive to detect those things. So need improvement. But then we need to know how in molecular, how this T cell work. Without knowing that, how can we find a good strategy to improve it? So that's basically the goal of our computational study. Okay, so let's first review how this kind of chimeric T cell receptor has been designed. Okay, so it's designed especially by mimic the native T cell receptor, how it works. It needs 2 signals for the native T cell receptor to activate. So first signal, okay, so here TCR is the T cell receptor. It's first need to recognize the antigen, and this present by the presenting complex. Okay, so the T cell receptor complex in our body is very complicated. You see, it basically has 1, 2, 3, 4, 5, 6, 8 protein complex, and it has this part recognize the signal, basically antigen, and then it activates this part, and this part ITAM is an immunoreceptor tyrosine-based activation motif. So Lily has the 6 tyrosine. When activated, it needs to be phosphorylated by a kinase called LCK. So then when bound, you somehow have to trigger the LCK to come here to do the phosphorylation, then it's activated. Okay, so the developer of the car borrowed this idea and to transfer the antibody here and to put some the linker and the transmembrane part, and then just cut this portion CD3 zeta domain put here. But amazingly, you put the signal antigen here, it's able to activate the T cell and to be cytotoxic for the tumor. So that's really a fantastic work. Okay, and people start to suggesting how it activated that part, because that's the central part. Okay, so people found out in that portion, in this CD3 zeta portion, well, it has tyrosine, so why those tyrosine? Okay, and so 6Y, and then it has some the polybasic radiosite, the lysine, arginine, which are the basic negative charge and positive charge. And then people suggesting those charge may play some role to interacting with the acidic and the negative charge one. So that basically in active state, and the CD3 zeta basically will bond to the membrane. Okay, then when you have the antigen, and it will bind and trigger conformation change, so those will expose to solvent. So then the LCK can come to do the phosphorylation. So that's activation. So they call this safety on-off model. And unfortunately, and this model developer uses a simple system, but when it's retesting in vitro and to do simple testing, so if this will work because they saw bridge, the people modify those basic radio into the aniline neutral radio. So then you will expect this interaction will remove. So this will stay in the solvent. When they stay in the solvent, LCK will come to do the phosphorylation. So you will say the phosphorylation rate higher. Unfortunately, the experimental data does not support. Multiple group report no, does not work. So that basically means this model at least not fully work. So we need got good understanding what's really going on. And another thing, this model does not explain why this bonding here can trigger broken the cell bridge and leaving this part go into the solvent. So there's no explanation for that. Okay, and okay, let's continue. Well, so the first generation CAR still not able to use in clinic because then people found very quickly that T cell will lose its cytotoxicity because it's not persistent. Okay, and so the developer for second generation of the CAR T looking for the second signal. So second signal in the wild type has many second signal, okay, and which makes the T cell activation more persistent. So the one they use this 41BB and CD3 zeta, okay. So they basically to cut the domain for CD137 and put here. So that's basically here. And then when put here, and so it's amazing. So reuse this transmembrane domain and reuse this antibody part. Well, it's able to make this T cell persistent. Okay, that's basically second generation of the CAR, and this is the one to save the life for the Emily. Okay, so how it work? Also, people suggesting it's not through the phosphorylation. It's kind of this is the native interaction. So native interaction is more about the molecular adapter. So when the activated is change conformation, allow its partner protein to bind. So partner protein is this one to bind, and once it binds, so you see recruiting the other protein, and then you have the signal transduction cascade going on. So appeared activation is a change conformation due to bond. Okay, those are the available information, but the detail we don't know. So that's basically motivates our group to study. So we pick a model system. So this is a CAR developed for targeting the solid tumor, which are breast cancer. And it shows a promising in vivo result for killing the human HER2 breast cancer in the mice model. Okay, so here's its architecture. Okay, and here I show you its architecture. It's basically use the antibody targeting the HER. HER is biomarker. Okay, this is the antigen HER. And then it pick the hinge. Hinge, this one, use the CD8A. It's also borrowed from the native T cell receptor complex. Okay, and then the transmembrane is also the CD8A domain. And then 41BB, we just mentioned, put here. CD3 zeta, and put here. Okay, and we use the AB refers antibody, HI refer to the hinge, and TM refer to transmembrane, and the CS refer to the co-stimulatory domain, and SR refers the signal domain. We will reuse those later. Okay, so you look at the solve structure. As I mentioned, the experimental approach not able to solve in the structure for this disorder part because those parts are flexible. And available structure are the order part. So this antibody, this antigen, yes, there's structure. Okay, so here we do some the bioinformatics study to detect which part are disorder. So this is the hinge transmembrane part. Transmembrane part is order part, is helices. But this hinge is disorder part. Okay, and then for this co-stimulatory domain, large portion are disorder. And then for this signal domain, large portion are disorder. Okay, so we have to use other methods to generate the starting structure. Okay, I mentioned the homogeneous modeling. So we can use homogeneous modeling. So you kind of can say, yes, they are disorders. have a lot of the conformation. Okay, so well, we need to start from somewhere place. So anyway, we just pick the model one to build our receptor structure. So here's what we built. Okay, so we have the crystal structure we can easily borrow, and for the other part, we just form our homology model to build this structure. Okay, so this is our model for this CAR-T, and then this is the antigen. Okay, so with the structure model, now we can use N-2 to simulate this system because this system is very big. Okay, I have removed the water, so this is some just detail about the molecular dynamic simulation. And then you kind of can see we have this antibody part and the membrane. And then here I highlight the tyrosine, which is supposed to be exposed solvent and ready for the phosphorylation when it's activated. Okay, so then we can build we call the whole system, basically including the antigen bond. And then we want to compare. Okay, so you say the simulation length is the microsecond, and we do the multiple simulation, make sure we got the statistical significance. Okay, for those, then let's look at the simulation result. See, can we learn something for the signal transverse membrane? Okay, so okay, so this is without the antigen. Without antigen, well, you kind of can see this portion, yes, it's try to associate the membrane, but it's not fully buried in the membrane. Okay, and this portion is appear to be more fixed, but this portion is much more flexible. Let's watch again to see if you see. So this portion is very flexible to search the partner. To me, it's kind of like search partner. It's kind of keep moving search partner. And this portion, the intracellular domain is much more fixed. Okay, so let's look at does behavior change when the bind with the antigen. Okay, immediately you realize when the bind with antigen, this portion are now more fixed. But when this fixed, this portion now become very flexible. Okay, so that's kind of different we observe, but that might explain the recognize the signal for the intracellular domain. So here's more quantitative analysis. Okay, so we just plot the center of those domain and to see what we just described. So for the APO system, you kind of can see, yes, each dot is the center of this domain. Yeah, it's something more kind of the space. And this part is kind of the height and more close to the membrane, and then it's more fixed. But when you bind, so we use this black dot to refer the antigen. Yes, now it's fixed, and this is moving around. Okay, so that might be the maximum for the signal domain to sense the one. Okay, so here we also do it for the costimulatory domain. Costimulatory domain don't have such a dramatic change on flexibility this part. And so we kind of think might be the more intra-domain conformation change for the costimulatory domain. Okay, but for the signal domain, and we clearly see it has both kind of more flexible and also have some inter-conformational change. So both inter-conformation change and intra-conformation change. Okay, so here's more detailed kind of analysis. Use the root mean square deviation, which characterize conformational change. Okay, so here we have the two systems. We have the APO system, which are this yellow color and blue color or hollow system. So you just need to notice the value are different. So which basically means the bonding. We started exactly same conformation, but after bonding with the antigen, and you see the conformation change. So this is bonding induced conformational change. And then we decompose that change into different domain, and that's the antibody domain. So you see the huge change, and the hinge part, and there's some change, and transmembrane there's small change, rigid part, and costimulatory part there are the difference, and signal domain, yeah, they are different. So bonding induced conformational change for signal part. Okay, so we use another measurement, root mean square fluctuation, which basically characterizes the flexibility of the different part of the protein. And then we kind of can say follow our expectation. So this is the transmembrane part. Okay, and the antibody and signal part has a big change. Okay, this is the comparison of the second structure, and you can see those two systems are different. Okay, so here's a kind of a key idea. So now we kind of thinking the activation mechanism is that it's bonding induce the conformational change, and which including the interdomain change, and also intradomain change allow it to change the conformation, allow the kinase to come here to bonding. And for this is our suggesting mechanism. So mechanism basically is to show that before it's a recognized antigen, it's a highly flexible, okay? And when the bond, it's fixed, but this side become highly flexible, and which will allow the kinase to bond here to do the phosphorylation. And this is pretty much follow the high school physics. And you can see here, the oscillating. So this oscillation can transduce signal to this side through this flexible string. Okay, so this is just mimic here. You can transduce that flexibility cross membrane through this bonding recognition and cross the membrane. So we believing this is the model. Okay, so with this model, how to improve the sensitivity selectivity? Here's the strategy. So we're looking, use that model, we're basically looking the nature. So nature use a lot of the antibody domain repeat, knows the kinase domain repeat. Now we hypothesis that the nature improves the sensitivity and the selectivity. So with this idea in our lab, now we try this idea. So we have the repeat on the signal on the antibody part, and we also want to try the repeat on the kinase domain. For this idea, good news, some group already developed the bispecific CAR-T with the two kinds of antibody domain to increase the selectivity. Okay, those are the model for the nature for this part. How the nature uses strategy to increase sensitivity, selectivity, and you can see yes, they follow the same architecture, use a flexible part to link those domain. Well, without the student, I was not able to present this kind of story to you. And so Stephanie, undergraduate student, start this project, and Maria, the graduate student, Lee are the graduate student, and they do the analysis, and Lee now do the experiment to verify our model. And here's my group, and our computational grant from the NTAN and the NIS grant and other grant. Thank you for your attention.